Good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Ensuring Microbiological Quality and Safety of B2B and Finished Products. My name is Sonia Hunt and it's my pleasure to be your X-Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streamings on xtalks.com. At this point, I would like to thank NIZO, who developed the content for this presentation. NIZO is world-leading in contract research and contract manufacturing with more than 70 years of experience and expertise. NIZO supports food and health companies in solving their challenges throughout the entire production chain. NIZO delivers development and application of innovations, resulting in gains in product quality, sustainability, cost effectiveness, and speed to market. Now, I'd like to introduce our speakers for today's event, or speakers rather. Marian Wells-Benick. Marian is a principal scientist in food safety. Her focus is on preventing and solving food safety and quality issues in foods together with NIZO's customers. This includes the control of undesirable bacteria in foods, which show an enormous diversity and can cause foodborne illness or spoilage. To provide solutions, the NIZO food safety team applies multidisciplinary approaches, including microbial and chemical risk assessments, troubleshooting activities, including whole genome sequencing approaches, challenging testing, and process validations. Our second speaker is Robin Islander. As a senior project manager at NIZO, Robin works on improving food and health with a focus on characterization of beneficial and harmful bacteria in silico and in vitro that are important for human well-being. In addition, a central theme of her work relates to spore-related issues in the food industry. In her work, she combines acquired knowledge and expertise as a research scientist with communication and organization skills for efficient research, increased understanding of scientific data, and solutions for customers. And now it is my pleasure to hand over the controls to our first speaker, and that is Marian. And Marian, when you're ready, you may begin.
Okay, thank you. So thanks once again, Sonia, and to all of our listeners, welcome to our webinar. We will start with a short introduction on safety and quality in food chain. In the presentation, we will focus on aspects related to R&D, particularly product design and safety, and on quality assurance, namely control and risk assessment. Let me see, sorry about this. It's not moving now. Yeah, here we go. So as you all know, food safety is a continuing challenge to the food industry, and it's essential to produce high quality safe foods, as food safety is a license to produce. Production of safe foods is really a prime requisite for public health, and of course it really prevents high cost due to loss of product, just to make sure that you get this right. It's also important that there is no recalls, because this may compromise the image and brands and consumer trust. New challenges may also be encountered due to new developments, for instance, the use of new ingredients, and I believe plant protein transition and the use of new plant ingredients may be an important component of this. In addition, new approaches may be needed to ensure food safety. So as Sonia already mentioned, NIZO is a contract research company, and among our expertise fields, NIZO helps to protect your image and brand by providing support on food safety and chain control. We take a broad approach to food safety, and provide interpretation of often complex and multidisciplinary results and data. We give meaning to this and provide hands-on solutions. This may be related to raw materials and ingredients. It may be related to product formulation and processing conditions and finished product properties. NIZO has 70 years of experience and our roots are in dairy, but over the last decade we've gained a lot of expertise and knowledge on many other products. Your challenges may be related to R&D, for instance, reformulation to meet consumer demands due to, for instance, the increased use of plant-based ingredients, but it may also relate to milder processing conditions that are applied to have a higher freshness of products, and it may relate to reduction of salt and sugar, for instance, or the reduction of additives. Your challenges may also relate to quality assurance with a focus on production of safe and finished products of high quality, as food safety, as I mentioned before, is a license to produce. So this brings us to the first poll question.
Well, thank you very much for that. Yes, it does bring us to our first poll question, and that will appear shortly on your screen. I will launch it now, and you should see it momentarily. Okay, and there it is. And the question is, in which part of the food chain control are you active? And you have two options here, research and development or quality assurance. I'll give about 30 seconds for this question here, and then I will close the polls because this is done in real time and I'll be able to share the results with you as soon as I close the polls. So your participation is very greatly appreciated. So if everyone could go ahead and cast your vote. It looks like we have about 75% of our participants have already casted their vote. I'll give another 5 seconds. Okay, and I'm going to close the polls now and share the results. And there's the results. 55% said research and development, and 45% said quality and assurance. Thank you so much for participating in that poll. And now I'm going to pass over the controls and the mic back to Marion. So Marion, when you're ready, you may begin.
Okay, thank you, Sonia. So looking at what NIZO does, we provide support in the food chain to ensure food safety and quality. And we have knowledge on the entire chain and apply a holistic approach to assess ingredients, but also formulation and processes and finished products. And we also have expertise on how these various components have an effect on each other. This can be assessed by a number of different services that we provide, and some of these are shown here. These are all relating to the analysis of ingredients, the modeling and optimization of product formulations and processing, and they are related to the analysis of finished product. We offer consulting and we offer troubleshooting advice. I think it's important to mention that we have our own laboratory facilities and we have a pilot plant. So we have a lot of expertise in the different areas. As I said before, microbiological challenges may relate to product design and stability in R&D or control and risk assessment in quality assurance. A typical pipeline that we use to assess microbiological safety in product in relation to design and stability consists of typically the following steps, and these are shown here. So this includes risk assessment and predictive modeling, the optimization of concept formulations and processes, and having done all that, we typically provide validations, and these are done in vitro. Usually we start with high throughput screening to actually be able to test a lot of different conditions, and ultimately we can test this in real product produced either by our customers or in our pilot plant, and we can perform challenge testing. I'm going to go through these different steps in a minute. Now, what may be the drivers for product redesign? And this is generally linked to consumer demands, but it may also be related to market access and distribution chains. So when you look, for instance, at consumer demands, the drivers may be healthier and fresh foods. But when you look at the distribution chain, it may actually be increased shelf life or longer shelf life. And the typical things that I mentioned before and that you may come across are, for instance, a reduction of sugar and salt and preservatives, and these may have side effects. For instance, the water activity may be affected, and this may then all of a sudden give problems in microbiological growth. The same applies to mild processing conditions, and this may actually reduce the microbiological inactivation or the microbial inactivation. Shelf life, another thing. When the shelf life is extended, all of a sudden bacteria may be able to grow to numbers that are undesirable. So clearly, the redesign of products and also the redesign of processing may have an impact on spoilers that you're seeing, on pathogens you're seeing, survival of pathogens and particularly spores by heating or inactivation processes may be changed, and changes to the distribution and storage conditions may have an impact. Last but not least, specifications on ingredients may be important, and what you may see here is that certain supplies of ingredients, when they are in short demand, sometimes specifications cannot be met. So these are all important aspects of food safety. And then we have another important trend that has an impact on product design and stability. And we believe that this transition from animal to plant protein is a very important one. Clearly, there is a wealth of knowledge on dairy, for instance, and this is one of the key products that NIZO has been working on historically. And what you see in dairy is that it's quite well known. The contaminants are quite well known. The levels are quite well known in raw milk, for instance. The product formulations companies are working with are very well established. There is a known impact of processing and known relevant specifications for ingredients. For plant-based proteins, however, this is really not the case. So what we are facing here often is that the contaminants and the levels thereof in ingredients are unknown. Product formulations are completely new. The impact of processing is unknown, and what you often see here is more fouling, for instance, because the properties of the proteins are different. And last but not least, we may be looking at new hazards and contaminants, and specifications of these important new hazards or contaminants may not be known. So we may not know how relevant certain organisms are. So this brings us to poll question 2.
Yes, it does, Marion. Thank you so much. And I will now encourage our audience members to participate in this interactive poll. It will appear shortly on your screen. There it is. And I'll give you about 35 seconds to complete this poll. And as you know, I will share the results with you right after I close the poll. And the question is, which challenges due to consumers' demands are you facing? You could select one, low sugar, low salt, clean label, mild processing, or protein transition. Looks like we have the majority of everyone voting now. I'll give a few more seconds, another 10 seconds on this. Your participation is greatly appreciated. 5 seconds. Perfect. Looks like we have a majority of everyone has voted now. So I'm going to close the polls and share the results. And here are the results from from this poll, which is which challenges due to consumer demands are you facing? 11% said low sugar, 6% said low salt, 44% said clean label, 14% said mild processing, and 25% said protein transitions. Thank you everyone for participating in that. Marion, I'll now pass over the controls and mic to you.
Okay, thanks again. So when we go a step further, so what are now typical steps that you can take to assess risks related to new products? And we typically start with identifying potential hazards and concerns. And for this, you can take a look at the composition of the product and ask yourself the question whether it supports growth. This can initially be done by predictive modeling. You can look at the different product characteristics such as acidity, water activity, antimicrobial presence, and storage conditions, for instance, the time and temperatures. And this can help with the identification of hazards. We typically do this by modeling potential growth of microbes. And of course, the focus here will be on pathogens, and there's a list of them here, vegetative organisms and spore formers, but we can also be paying attention to spoilers. You can, of course, also look at the process and ask yourself the question, what the effects are not only to product quality, but also contaminants and microbes. So here you can look at things like processing units, heating, the time temperatures used, residence times and temperatures, and fouling potential. And this helps to pinpoint potential hazards here. So again, here we are looking at modeling of growth, but certainly also, and this is different from the product side, we look at inactivation and possibly also removal of microbes. Again, of course, pathogens are important, but here I would really like to stress the difference between the vegetative cells, which are typically inactivated quite easily by heat, and spores of especially the pathogens Bacillus cereus, Clostridium botulinum, and Clostridium perfringens. And last but not least, of course, spoilers will also be a concern. It will be needless to say that processing conditions and product composition have dependencies, and these need optimizing, and I think it's important to look at all the different aspects. So now let me give you an example of how product composition and storage may influence the types of microbes that are of concern. What we typically first look at is whether a product allows for growth or whether it needs further adjustment. For this, we assess the product composition in light of boundaries that are present for the growth of particularly pathogens, and these are mainly water activity and pH. For instance, when the pH is below, let's say 4, a lot of pathogens will not even grow. So this is an intrinsic property of a product that is important for microbial stability. If the product does allow growth, we will assess how fast this may occur at different temperatures and whether the critical levels in a product may be reached at the time of shelf life. The images here on your right, they show an example of a product with a low water activity on the left and a higher water activity on the right. And at the top you see growth conditions at 21 degrees centigrade, so ambient conditions. At the bottom, 7 degrees, refrigerated. These images are just illustrating that depending on, for instance, the water activity or the temperatures, different risk organisms may be very abundant or may be very clear. And this is, of course, logical, and it is linked to the intrinsic properties that bacteria have. I would like to emphasize that it's important to do this in my view, before even developing a product. I think it's important to take these kind of considerations into account when you're developing a new product, because you may already get to a no-go zone because of certain pathogens that you may not be able to eliminate. So by taking this as an initial approach, just a modeling approach, we are able to generate a rapid identification of the main microbiological food safety risks in products during storage, in principle all the way through to the shelf life conditions and the use by consumers, with a focus on the critical contaminants. So this gives a fast insight in the risk microorganisms, and it allows for a targeted approach toward ensuring safety. So clearly, heat treatment in processes also have a major impact on contaminants. And here, as I mentioned before, we tend to distinguish between vegetative organisms and bacteria that can produce spores, because spores may survive and they may later, after they survive processing, they may germinate and growth. And Robin will talk a bit more about that later. Here it's important to assess the efficacy of inactivation, and we do this typically by modeling, again, for different risk organisms, particularly spoilers and pathogens. What is also important to mention here is that we take variability into account. So what you typically see is that there's variability between different species. For instance, Bacillus cereus may have different properties compared to Clostridium perfringens. But what we also know is that you can get a lot of variability between strains of a certain species, and we can take that into account as well. So we can calculate worst and best case scenarios or average scenarios. So this approach allows for fast assessment of heat and activation of spores by treatment of products. So based on initial modeling, it's generally clear whether further adjustments in the product, for instance, product formulations are needed, or that adjustments in processing are needed. In that case, we can start out with a desk study. And we do this to see if there's alternative strategies that may be applicable. This actually requires an assessment of the efficacy of compounds, and we also take applicable regulations into account. We also look at the potential impact of taste and product characteristics, because certain compounds may have very undesirable flavor and taste effects. So here you can actually see a number of potential solutions, and this may be in the area of natural antimicrobials, for instance, bactericins or essential oils, which actually have quite a strong taste and flavor effect. But it may also be organic acids, ferments, and clean label solutions. And of course, we can also look at storage and shelf life adjustments. And we typically use hurdle approaches and use combinatorial approaches here. So this provides an assessment of the alternative concept formulations to target identified risk organisms. To actually evaluate the solutions, we have high throughput facilities at NIZO. And this is a very, very versatile platform that we have in place. We can use this to assess the growth of different microorganisms in real time, in real products, and we use CFU counting with up to 3,000 different conditions simultaneously. We can do this aerobically and anaerobically, and what is important here is that we have a very large culture collection with pathogens and spoilers that we can employ, so we can select critical and important organisms. This is a very powerful tool to assess whether potential solutions for control really work before going to very costly challenge tests in real product. But of course, ultimately, the proof is in the pudding. And for this, we can test real product, either supplied to us by our customers or made in our pilot plant, where we have aseptic filling capacity in test size aliquots. These can be used in challenge tests for microbes of concern. The spike products can be stored under relevant shelf life regimes, and this provides the ultimate answer on safety and quality in practice. We provide expert advice on study design and interpretation, and we provide translation to practical solutions. So hereby, we can validate the screening outcomes in actual products and meeting consumer demands while maintaining product safety. So to sum up this part of the presentation, I would like to stress that all parts of the chain are important to the production of high quality safe foods. This starts with the ingredients, includes formulation, processing, and the storage conditions of finished product. But even so, unexpected cases of spoilage and safety may arise, and it may be needed to reassess the chain starting from ingredients. Robin will talk about that in the QA, about the QA issues in the next part of the talk. But before doing so, I would like to end with another poll question.
Yes, yes, we got another poll question. Thank you so much, Marianne. Just as we did before, audience members, your participation would be greatly appreciated. I have now launched the poll question. I'll give about 35 seconds for this. And the question you have today is, what is your main area of interest in the food production chain? I'll give you a few moments to answer that. Your options are ingredients, product formulations, processing, finished product, and product release. I'll give a couple of seconds. Hopefully everybody will be participating in this poll. Okay, and now I'm going to close the poll and share the results. And here are the results from the last poll. 29% said ingredients, 26% said product formulations, 16% said processing, and 29% said finished product and product release. And now, thank you very much for participating with us in that poll. I'm now going to pass over the controls to our next speaker, and that is Robin. Robin, you may begin when you are ready.
Thank you, Sonia. So this is a new microphone. I hope that the sounds are okay. Yeah, so let me go ahead and tell you more about our quality assurance pipeline for control of your finished product and risk assessment throughout the chain, starting with product release or root cause analysis in case of a spoiled product, the value of microbial characterization in risk assessment, and also the importance of reliable enumeration methods, but also reaching harmonization in the market on the methods applied, and how you can use data of microbial properties that can help in optimization of processes. So when you're dealing with a spoiled product, it is of the utmost importance to understand the severity of the contamination that you're dealing with. For instance, you could be dealing with a spore former or a biofilm former. In case of the latter, the cells will be able to attach to processing equipment and grow and even produce spores in time. So this will lead to recontaminations of your product. In case of a pathogen, safety may be compromised. So this is a microbial hazard in case of toxin production. Also, it would be good to obtain some insight on potential resistance to understand the efficacy of mitigation strategies that are applied, such as application of acids or heat, antimicrobials or cleaning agents. So at NIZO, we offer a rapid microbial identification service, which is based on 16S profiling. What we do is we isolate the DNA directly from the contaminated product. So this is a culture-independent approach. That means that identification is still possible even if cells are no longer culturable. Additional to the identification, we also offer expert interpretation on the actual identification applied. So that is phylogenetic relations to previous contaminations, the classification of safety of the microbe at hand, and the natural habitat. So with this identification, you get rapid insight in the main bacterial contaminants of your spoiled product using DNA sequence analysis, including also background information in relation to food safety and the possible root cause. Of course, this can be continued by a more in-depth characterization using whole genome sequencing to allow for strain-to-strain comparisons and more in-depth track and trace. And at NIZO, we also provide consultancy on what would be the best next steps and optimization of your processes. So what could be the potential issues that you are facing when you're actually dealing with microbial contaminants of products? So on the one hand, of course, it could be pathogens. Once that has been identified, it's very important to know whether the strain that has been identified actually has presence of genes encoding for toxins. So we can do in silico screenings for the presence and absence of such genes. Also, it's important to then assess whether these toxins are actually produced in products. So we have chemical detection assays, for instance, for cerulide and licanicin, which are similar to the Staphylococcus enterotoxin, quite heat-stable toxins, meaning that they remain a hazard once they are produced in the product, even after heating. Less stable is the botulinum toxin, but this is one of the most potent toxins known to man, so this really compromises safety. If you're dealing with contamination by pathogens, safety may be compromised, leading to product recalls and damage of your image, brand, and consumer trust. If you are dealing with spore formers as a contaminant, then the most important issue there is their heat resistance. How much that heat resistance is really depends on the matrix in which the spores reside. So there you can use the information of D and Z values to really predict the efficacy of your thermal processing based on heat inactivation and kinetics databases for predictive modeling to see how efficient those processes really are in inactivating the spores. When you're dealing with contamination by spores, then the main issues are mainly product loss and market complaints, but also you will need to reassess the ingredients, the supplier of your ingredients, agreements that you make based on specifications, and also how your processes are designed. So let me give you a little bit more information about these bacterial spores. Here you see a movie running, and in this movie you can see spores being produced by the bacterial cells, which is a survival strategy to limiting nutrients. In this dormant and metabolically inactive form, spores don't really do anything, so they don't cause issues in this state, but they are extremely resistant against all sorts of stresses due to a very low water content in the spore core and many protective layers around it. They need to germinate and grow in your product before they cause spoilage and safety issues. This is a process that starts when conditions improve for the spore, like the availability of nutrients or nice temperatures for growth. Once they germinate, they lose their resistance properties. But a strategy to first have spores germinate and then apply milder heat treatments won't really work. You can see that on the next movie, because not all of the spores will respond immediately at the same time. As you can see here, one spore is not germinating at all. These spores will eventually germinate and cause issues in your product. So to give you some more information about spores, it's an ultimate survival mechanism of these bacteria, and it's important to know that they're everywhere, from the farm, ending up on the udders of the cow, into the raw milk, into the milk tank, and in your processing equipment, and eventually in your finished product. So it's not so much a sense of how to prevent them from being present, but more how to deal with them. Bacterial spores easily survive pasteurization, and in some cases they can even survive sterilization. And some of these spore formers like warmer temperatures, and they are able to attach to processing equipment, so they can thrive in evaporators, for instance. As Marion already said, some of these spore formers produce also toxins, so they're also pathogens. And it's important to realize that spore-forming bacteria express a high degree of strain-to-strain variability. So it's not only species-dependent variability, but also between strains of the same species. So especially with bacterial spores, it is not a focus to how to prevent them from being present, but rather how to reliably detect them and interpret spore counts for the degree of risk assessment, spoilage in products. Normally, classical plating methods for that are used, but they often lead to underestimations. The recovery medium, for instance, really needs to support the germination and outgrowth of the organisms. Otherwise, you're just not seeing it and you may think it's not present. Pre-treatments in the method may affect microbes that are present in the sample. And classical plating methods have a high limit of detection because dilution is required. So it doesn't really allow for the detection of very low numbers. Those low numbers of spores can still cause issues in finished products. Classical plating methods also do not distinguish between different species or strains, and they do have varying properties, so that complicates risk assessment. Other challenges that are seen is that standard methods may be available, but they're not always practical and globally applied. We know from experience that different methods to detect spores really yield different results. So how to then interpret them and make agreements? This leads to disputes in the market of business to business on how to interpret the results and set relevant specifications. So a couple of years ago, we started an industrial consortium with a couple of other companies with the main goal to develop a simple, relevant, and practical method for the detection of highly heat-resistant spores in milk powders as a practical alternative to the available ISO standard, which requires heating at 106 degrees. What we found is that tryptic soy agar is recommended recovery medium over the plate count milk agar that is stated in the ISO method, simply because the tryptic soy agar medium supported growth of all of the bacterial spore formers that were tested in our study. Also, we showed that heating at 100 degrees for 30 minutes instead of 106 degrees Celsius is possible as an alternative method, but specifications should be increased tenfold. Classical plating methods offer about 2/3 reliability and prediction of spoilage of the reconstituted UHT milk using the same milk powders, which means that 1/3 is still unpredictable based on this variability that we see between the different species and strains of the same species. So this work has been published in case you would like to know more about it. We continue the consortium as a follow-up to focus on cocoa powders, since these also form additional challenges compared to milk powders. For instance, cocoa powder is known to have an antimicrobial effect, so again, you can have further underestimations of the counts on plates. Due to its dark color, especially alkalized powder, it's sometimes difficult to distinguish single colonies on agar plates. And cocoa powder specifically contains a lot of bacillus species, which are known to swarm. So therefore, single colonies may not be detected, again leading to underestimation. And cocoa powder especially is notorious for its poor hydration properties. So if you're not hydrating your sample properly, you will get dry heat exposure to the bacteria that are present. And it's known that spores in particular are very resistant against dry heat, so they will always survive. In an industrial consortium with cocoa producers and cocoa buyers, we optimize this detection method, which has now been submitted for publication. And then it's important to mention that we're now this year also starting a new consortium that is directed towards microbial safety of plant protein ingredients. As Marion already said, there's still many unknowns when it comes to these proteins. We don't know their microbial contaminants and their properties. We don't know what the influence of plant protein properties are on the efficacy and reliability of microbial detection methods, or what the influence of processing is on the actual quality of the plant protein. When you're dealing with novel product formulations with these new ingredients, you get an increased risk of spoilage in finished products. So we're starting this novel consortium, and we're applying for funding to also be able to make a significant contribution in ensuring microbial safety within this protein transition trend. Then the last example that I would like to show in this webinar, I would like to highlight how NIZO makes use of knowledge and data of bacterial properties, such as spore heat resistance, to help you optimize your thermal processing strategies for inactivation. As I mentioned before, heat resistance of spores is greatly dependent on the matrix in which they reside. So at NIZO, we determine D and Z values of the spores of interest and of reference strain directly in the food product. This data is then plotted against an extensive database on spore heat inactivation kinetics to show how it relates to known inactivation kinetics of related strains. Using these model predictions, we can define the most suitable settings for heat inactivation during processing. So through such an approach, you will be able to make a substantiated decision for your thermal process design based on inactivation of identified spores. We can help you recommend on efficient time-temperature combinations for log reduction in your processes, and if required, also help you validate such processes design on a lab and a pilot scale. In view of time, I don't have time to explain the figure on the right with all of the different colors, but the main message there is that there's a great variability in different strains of various different species when it comes to heat inactivation. So to summarize this webinar, we have shown and focused on the importance of tackling the entire chain to ensure microbial safety of your product, from ingredients to finished product, and shown approaches and solutions that NIZO has to offer to support you in that. When looking at ingredients, we can help you identify and characterize both microbial and chemical contaminants and investigate the efficacy of detection methods for optimization, increased reliability, and helping you in finding harmonization with your suppliers or buyers. By evaluating the link between analytical outcomes of ingredients and the risk of spoilage of product after processing, we can furthermore assist in setting relevant specifications. When going into novel product formulation, we can help assess and optimize key characteristics of products and processes to predict either the potential for microbial growth or inactivation using mathematic modeling. And we can apply high throughput screening for fast and cost-efficient assessment of most suitable conditions to validate findings, both lab and pilot scale. And then for finished products, we can assess product shelf life validate inhibition and inactivation of microbes, but also identify and characterize organisms causing spoilage, both in silico and in vitro, to support you in root cause analysis, track and trace, and optimization strategies for the production pipeline to prevent such contaminations in the future. So through a multidisciplinary approach, tackling the entire chain, we help to validate safety. And at NIZO, we have a great team of experts to provide both consultancy on and expert interpretations of outcomes. And with that, I really would like to thank you for attending this webinar, and we're happy to take any questions.
Thank you very much, Robin. I hope everybody enjoyed the webinar. Now that brings us to the question and answer portion of the webinar. Robin, are you ready? I have a couple of questions here that have already come through from our audience. Are you ready to take them?
Yes.
Okay, so here's our first question. You mentioned rapid identification of main bacterial contaminants in spoiled through 16S sequencing, but does that give you sufficient resolution for track and trace?
Well, to answer that question, no, it does not. It gives you at least information on the genus and sometimes on the species level, but that may not be enough for the real track and trace if you want to go into strain-specific differences. So if you want to know whether you're actually dealing with the same strain that keeps contaminating your product, you really need to go into further genetic identification. So whole genome sequencing can offer a good solution for that. And then, of course, combined with bio-IT efforts to really identify where the similarities and differences are when it comes to previous contaminations.
Okay, perfect. Thank you so much for that. We have a couple more questions lined up here. Here's the next one. You mentioned something about new or unknown hazards and contaminants present in plant proteins. What do you expect there?
So at the moment, we don't know too much about the contaminants of plant protein ingredients. We know there's definitely bacterial contaminants as well as fungal or yeast infections that may take place. So it's difficult to say at this point which those are. There's still a lot of gaps in our knowledge when it comes to those. Definitely, we expect spore formers to be present in many of these plant-based proteins, similar to what we found for cocoa powder, for instance.
Thank you for that one. We have another question right now from our audience, and this one is, earlier you mentioned very briefly your experience in fermentation strategies for biocontrol. Can you elaborate?
So when we're looking at fermentation control, I hope there's not too much of an echo on the line. What we have here at NIZO is an extensive expertise on fermentation, both in the area of dairy and on plant-based ingredients. So this can be applied to, for instance, provide ferments, but you can also think of production, for instance, on cheeses or yogurts. And what is very important there is that, for instance, the acidification rates are fast enough to prevent other organisms from growing. Another issue may be that there is competition of often the lactic acid bacteria with undesirable microbial contaminants, and this can actually be solved by selecting the right cultures and the right conditions in different products.
Thank you for that. And now we're going to go on to our next question from our audience, and that is, would you be able to give examples of the spores resisting the evaporator process?
Sure. Yeah, so what you see in evaporators, typically these are run at conditions of 55 degrees centigrade, and there are a lot of bacteria that can grow under those temperatures and those conditions. So the types of organisms you typically see there are Bacillus species such as Bacillus licheniformis, but also the obligate thermophilic organisms such as Geobacillus species. So these species, they are able to grow under those conditions, and in addition, they are able to produce spores while they are going through the evaporator, and this may then end up, the spores may ultimately end up in the milk powders that are produced that way, or if it's not a milk powder, but other types of evaporated streams the spores may end up in the powders that are evaporated and usually often dried after that. But it may also be a problem in evaporated products as such that are subsequently canned, for instance.
Thank you. Oh, that's great. Thank you so much for that. Now we have our next question, and that question is, on a previous slide that you showed, what does it mean that spec needs to increase tenfold?
Yes, so Sonja, this is Robin. I can answer that question.
Thank you.
So what we've seen is a linear correlation between the count that we found for spores in milk powders when we apply the heat treatment of 30 minutes at 106 degrees Celsius and 30 minutes at 100 degrees Celsius. So there we could find a linear correlation that every time the counts that we found were tenfold higher when you are applying a heat treatment of 100 degrees Celsius for 30 minutes. Obviously, 106 degrees Celsius is a much higher heat treatment for these spores, so only the very highly heat-resistant spores will survive and your counts will be lower. And due to this linear correlation that we found in the counts when we compared both conditions, we can state that if you are using, for instance, 100 CFU per gram as a specification when you would apply the method of 106 degrees for 30 minutes, then you can alternatively apply a method using a heat treatment at 100 degrees Celsius for 30 minutes, but your specifications need to increase to 1,000 CFU per gram.
Okay.
I hope this answers the question.
Okay, I'll go on to the next question then. The next question we have is, can you indicate what the advantages and disadvantages are of your high throughput screening facilities? I'll say that one more time. Can you indicate what the advantages and disadvantages are of your high throughput screening facilities?
Sure, I will take that question. Can you hear me okay?
Yep, you sound great.
Okay.
Thank you.
So the high throughput facilities that we have, they allow for the measurement of CFUs in real products. These can be liquid products, but it can also be solid products. For instance, we have a model system for micro cheeses, but we also have a system where we can analyze liquid dairy products. But in principle, we can apply this to any product that is of interest. What is one of the disadvantages of this system is that we cannot work with very low contamination levels in challenge tests. So we typically have a level of 10 to the 4 as a starting inoculum, and this has to do with the limit of detection in the system, because we are working with such low volumes of typically 100 micrograms or 100 microliters. However, the major advantage is that we can screen a lot of different conditions. So when we make, let's say, a matrix of combinations of antimicrobial compounds or different testing conditions or different formulations of products, we can really use this to screen the efficacy of combined approaches against a large number of different pathogenic species or spoilage forming organisms that may be relevant to a particular product. So yeah, I think one of the main other advantages that we have in this system is that we can use turbid products, and this is often a problem in foods because you have you don't have a clear fluid, so you cannot assess growth or no growth by just looking at optical densities of cultures. You really have to do CFU plating, and this is a very strong feature of this platform.
Okay, perfect. Thank you. Yeah, no problem. Thank you. All right, here's our next question. Why TSA is good media for spore forming testing than PCM?
Yeah, then PCMA. So that is plate count milk agar or tryptic soy agar. This is Robin. Let me answer that one for you. So what we've done in this spores consortium on milk powders is we've tested 6 different classical plating methods using different types of media on about 40 different strains of bacterial spore formers of various different species. I think it was 16 different species in total. So we tested Geobacillus and Oxybacillus, bacillus species, and we really looked at the recovery of these strains on all of these different media. And what we found is that TSA was able to support growth of all of these strains, of all of these species, whereas PCMA did not. So I can't quite remember. I think it was Bacillus sporogeny. I'm not quite sure which one it was, but at least one or two of the species was not supported by PCMA. So in that sense, you may get underestimations in the counts of the spores actually present in your milk powder ingredient. Therefore, we recommend to use TSA as a cultivation medium.
Okay, thank you for that. We have another question. I think we have a time. Yeah, we have about 5 minutes left here, so we can go for a couple more questions. Here's one more here. Did NIZO also give consultant assistance to help evaluate the stability studies of the finished products, like evaluate all the data during the shelf life to understand why this vitamin is suffering more degradation than this other, or why this mineral is appearing during time? In T0, it has some value, but in T6, it's about 3 times more.
I'm not entirely sure what this question relates to, but I believe it has to do with the presence or absence of vitamins and how that may affect the growth of particular species. This is something we do see. Sometimes you have bacterial species that have quite strict requirements for growth, and the presence or absence of vitamins may really influence the outgrowth of particular organisms. I'm not quite certain what the question was, but it may also be relevant that certain vitamins are produced by other bacteria that are present, and this may then support growth of species that are also present.
Well, thank you very much for those questions. We have reached the end of the question and answer portion of the webinar. If we couldn't attend to your questions, the team at NIZO may follow up with you after this presentation. If you have further questions, direct them to the email address on your screen. In today's webinar, you will be receiving a follow-up email with X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, I'm about to send you a link in your chat box, and you'll be able to view the recording of this event at that link. And also share this link with your colleagues who were not able to attend today's webinar. They can also register for the recording as well. So I encourage you to do that. Now, please join us in thanking our speakers, Marion and Robin, for that very insightful presentation. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, take care and bye for now.